So thank you very much for this opportunity to present my project on the effect of silver nanoparticles on the viability of bacteria, fungi, aquatic organisms, and plants. Many consumer products today contain silver nanoparticles, which are advertised to have antibacterial or antifungal properties that prevent bacteria from growing in everyday items that we use. Silver nanoparticles can attach to and alter the bacterial membrane. They can penetrate inside the bacterial cell, causing harm to important proteins and processes such as ATP production and DNA replication, which can cause cytotoxicity or cell death. When silver nanoparticles in personal care products wash off the skin, they enter into the environment with unknown effects, causing them to giving them the potential to cause unpredictable consequences for ecological health. I asked what concentration of silver nanoparticles has a cytotoxic effect on the bacteria E. coli and the fungi Saccharomyces cerevisiae. I questioned what happens if silver nanoparticles get into the water. How do aquatic organisms such as Daphnia magna, the water flea, and plants such as Vigna radiata or the mung bean respond to the exposure of different concentrations of nanosilver? My hypothesis is if higher concentrations of silver nanoparticles are used to treat these organisms, then more cell death will occur because higher concentrations of nanosilver are more cytotoxic.
This is a 7th grader. Hot damn.
E. coli bacteria were plated on soft nutrient agar petri dishes. Immediately after plating, 6 millimeter sterile discs soaked in a specific concentration of a silver nanoparticle solution and were then placed on agar plates and incubated at 37 degrees Celsius. The diameter of the zones of inhibition increased as the nanosilver concentration increased. I repeated the same experiment, but this time I soaked sterile discs into different consumer products containing silver nanoparticles. There was clear zones of inhibition with each product tested at 48 hours after incubation. I repeated the same experiment with the fungi Saccharomyces cerevisiae and got very similar results for both the nanosilver particle dose response and the consumer product testing. This experiment tested the effect of silver nanoparticles on the viability of Daphnia magna. 10 Daphnia magna were placed in a 20-ounce cup containing increasing concentrations of nanosilver mixed in pond water at room temperature. The number of living and dead water fleas were counted after treatment with a silver nanoparticle solution. The percentage of dead Daphnia was plotted for each nanosilver concentration and the control over exposure time. There are more dead Daphnia at higher nanosilver concentrations, and more cell death occurred after longer incubation times. My last experiment tested the effect of silver nanoparticles on mung bean seed germination. Mung beans were placed into different jars containing increasing concentrations of nanosilver. Mung bean seeds germinated into seedlings at lower nanosilver concentrations, and as the concentration of nanosilver increased, the seedling formation was significantly slowed. At the highest nanosilver concentration, the mung bean did not germinate out of its seed coat. In conclusions, the results from these experiments treating E. coli, S. cerevisiae, Daphnia magna, and Vigna radiata with increasing concentrations of silver nanoparticles supported the hypothesis that higher concentrations of silver nanoparticles cause more cell death because higher concentrations of nanosilver are more cytotoxic. In addition to confirming the antibacterial and antifungal properties of silver nanoparticles, these studies provided important information on the harmful environmental effects of silver nanoparticles on plants and aquatic organisms. The same antibacterial properties that make silver nanoparticles useful in consumer products may also cause the accidental killing of beneficial microbes that play critical roles in ecosystems and help treat wastewater. Therefore, the potential negative environmental effects of silver nanoparticles should be carefully studied and weighed against their environmental impact to control hazards associated with widespread use.
Well, thank